The last topic that we're going to cover in Unit 3 is biotechnology. Biotechnology involves the modification of genetic material in living organisms or cells. Cloning involves extracting DNA from a host cell and implanting it into another type of cell. Reproductive cloning allows the creation of an entirely new organism, while molecular cloning creates copies of a particular gene. The genetic code is universal, so a gene that codes for insulin in a human will also code for insulin in a bacteria. Bacteria contain extra chromosomal pieces of DNA. By taking the desired gene for insulin or another product and inserting that into the plasmid, we now have recombinant DNA, the combination of DNA from two different sources. This slide demonstrates how recombinant DNA technology can be performed. Genes for particular protein products such as insulin can be extracted from human cells. The slide shows the insulin gene can then be injected into a plasmid, and when the plasmid is incorporated back into the bacterial cell, the bacteria will produce insulin. Pituitary dwarfs of individuals whose dwarfism is due to the absence of human growth hormone. A federal study was conducted in 1985, and this found that out of 7,700 people in the United States who received cadaver-derived human growth hormone, 26 died of CJD. CJD is a human derivative form of mad cow disease. It's possible that more deaths are still likely to occur, as it takes more than 30 years for CJD to develop. Today, individuals that receive human growth hormone that's been cloned into bacteria. Human growth hormone increases the length of your long bones. Muscles need to grow to keep up with bone length increase. It also regulates your metabolism. It is sometimes used by athletes as they claim that it increases their recovery after injury. Human growth hormone and insulin are just two of the products that can be produced using recombinant DNA technology. Transgenic organisms are organisms that have had genes added to produce new characteristics. Transgenic organisms include bacteria, plants, and animals. Transgenic plants are also referred to as being GMOs, a genetically modified organism. Some of the characteristics that are added to transgenic plants include those that enable increased crop yield, resistance to drought and freezing. There are traits that are added to decrease the use of pesticides, increase the nutritional component of the plant, as well as increase its shelf life. Corn is a major agricultural crop that is often modified through plant biotechnology. Plants can be genetically modified to make their own insecticide. A gene from bacteria is inserted into crops that produces an endotoxin that kills pests. Other types of transgenic plants are resistant to Roundup. The herbicide Roundup can then be readily used and not affect the crop yield. This process is patented, and therefore the seeds cost more. The use of these crops may select for a few insects that are resistant. These resistant insects could breed together and produce a resistant strain. Naturally produced insecticides may also transfer to other plants. Bt corn is a corn plant that has been biologically engineered with a gene that codes for a protein lethal to the European corn borer. This gene is isolated from a genus of bacillus and is commonly used as a biological pesticide. However, this biological agent will drift from the corn plants to the surrounding milkweed, where it's been found to be harmful to monarch butterflies. While Roundup-ready crops initially made farmers giddy, there are now some types of weeds that are Roundup resistant. The wheat industry has long been resistant to any type of genetic modification. However, in Oregon, some genetically modified wheat was mysteriously found. Let's look at some transgenic animals. By inserting growth hormone genes, we've enabled the production of larger fish, cows, and pigs. Farming is a term used to describe the use of transgenic farm animals to produce pharmaceuticals in their milk. These types of genes only function in the mammary glands. An example of this is TPA, tissue plasminogen activator. This can be used in the treatment of strokes. The way transgenic animal is produced is the desired gene is injected into the animal embryo, and that embryo is then implanted into its mother. Now we're going to look at some animals that have been cloned. In reproductive cloning, a body cell or somatic cell is obtained from the organism to be cloned. The nucleus is removed and introduced into an N-nucleated egg. An N-nucleated egg is an egg that's been stripped of his nucleus. This egg is then implanted into a female of the species and produces an identical genotype of the adult donor. How is this possible? Every cell in your body receives a copy of all of your genes. So therefore, every cell in your body has the potential to become a complete organism. This diagram shows how the nucleus is removed from the somatic or body cell of the organism to be cloned. It is then fused with an egg that's had its nucleus removed and discarded. The cells are cultured until an embryo is formed. The embryo is implanted into a surrogate mother. After that, the clone of the original organism is produced. Does this sound like something out of science fiction? Plants are routinely cloned. As with plants, the desired endpoint is to have an individual genetically identical to its original. It was thought for a long time that the cloning of adult animals was impossible. This brings us to a famous clone, Dolly the sheep. Dolly the sheep died when she was 6 years old. At this time, she was found to have progressive lung disease and arthritis in her back limbs. These type of ailments usually only affect older sheep. The reasoning for this is that genetically, when she was born, she was already 6 years old. Her DNA was 6 years old, and it's theorized that this might have led to her early death. The success rate in the generation of Dolly the sheep was very low. Scientists started out with 277 cell fusions. That's where they took the nucleus of the adult sheep and added it to the N-nucleated leg. Out of these 277 cell fusions, only 29 actually began to grow as embryos. 13 ewes were implanted with these 29 embryos, and only 1 sheep became pregnant. The success rate therefore being about 0.36%. Human cloning is illegal, as well as it doesn't work. In the process of creating the N-nucleated egg, there are some proteins essential for cell reproduction that are stripped out when the egg nucleus is removed. Pet bank through the genetic savings and clone is a way to clone your obsessional pet. It costs about $1,395 initially, with an annual storage fee of $150. The entire cost of cloning your pet is about $32,000. And as far as appearance, having the same DNA as in one calico cat doesn't necessarily produce the same coat pattern in the cloned calico cat, and this company has had a number of lawsuits. In therapeutic cloning, the desired endpoint is to generate mature cells to learn more about cell specialization. Also to use cloned cells to treat human illnesses. This would be a way of treating spinal cord injuries or damage to the central nervous system done due to strokes. Therapeutic cloning can be carried out in several ways. First, embryonic stem cells can be used, but there are some ethical concerns and there's currently restrictions on the number of embryonic stem cell lines. Adults have some stem cells in their bodies. There are some muscle stem cells as well as those found in the bone marrow. But these are very limited in the number of cells that they can become. The research in adult stem cells is to find ways to overcome these limitations. The desired outcome would be, if you had some type of injury, to be able to take one of your body cells, diffuse it with an egg, to grow tissue and culture, and then cause these tissues to become specialized and to generate new nervous tissue, blood tissue, or muscle tissue. In the Netherlands, researchers were able to grow in culture the stem cells of 2 live cows and actually created a hamburger known as the Frankenburger. It was not, however, found to be very tasty as it consisted of only muscle tissue, and one of the components of hamburgers that brings about the particular flavor is the presence of fats.